This is Juan Mendez, and here is my photosynthesis video project. So before we get into the real nitty-gritty of photosynthesis, the real reactions, it's important to understand a few central concepts, starting with the electromagnetic spectrum. So what is it? Well, the electromagnetic spectrum displays a vast range of radiation in the universe. Radiation sounds kind of scary, but remember that all radiation is just the emission or transmission of energy in the forms of waves or particles through space. So the spectrum you see here lays out different types of wave radiation, looking sort of like a number line. And like a number line, the properties of each point are different, and the line as a whole follows a pattern. You can see them all here, the numbers, quote unquote, as gamma rays, X-rays, ultraviolet waves, visible light waves, infrared waves, microwaves, and radio waves. Visible light is really going to be the focus here in this video about photosynthesis. However, I want to touch on a few things first. An important factor of these different types of wave radiation is their wavelength, which you can see here. And defined, let's pull it up, is the distance from one wave peak to the next. And so you'll see that radio waves have really the longest wavelength. Conversely, you look at gamma rays, they have the shortest wavelength. And this sort of corresponds with energy as well, inversely. So longer wavelength, lower energy, as it says here. Shorter wavelength, higher energy. Visible light, which is going to be our focus, really represents a small fraction of the electromagnetic spectrum. It has a variety of wavelengths ranging from 380 to 760 nanometers. And so what we'll see is that the energy of visible light is used in photosynthesis. The reason this part of the electromagnetic spectrum is called the visible spectrum is because we as humans are actually able to see it. All of the colors of the rainbow are included in the visible spectrum, with violet having the shortest wavelength and the red the longest. The sun emits this visible radiation, which plays a key role in photosynthesis. Now, let's delve a bit more into light in the visible spectrum. Photons are important when discussing visible light and radiation. And what they are, as you can see here, are small particles or packets of energy that make up light. Photons are sort of like the monomer or building blocks of light. Knowledge of photons is also important in discussing the following question, which is, why visible light? Why am I talking about focusing on visible light, and what is its importance in photosynthesis? What differentiates it from other wavelengths of radiation? Visible light is important because it has a few key and unique properties. Visible light radiation excites certain types of biological molecules. And what I mean by excites is that it moves electrons in these molecules into higher energy levels. This picture demonstrates what I'm talking about. As you can see, when a photon of visible light radiation hits this atom, which we know is a hydrogen atom as it has one proton and one electron, when it hits the electron jumps up a ring or an energy level. It's sort of like this electron gets slapped by this proton. It's like, I really don't appreciate that, and it moves in irritation. Visible light is the only type of radiation that can do this. It's the only type of radiation that can slap the electrons. Radiation with longer or shorter wavelengths than the visible light spectrum do not have the same effect, as either they have too much energy, in the case of the shorter, which disrupts the bonds of molecules, or they have too little, in the case of the longer, not enough to excite the electrons. This really demonstrates why visible light is so important in photosynthesis, because it has just the right amount of energy to make this change in molecules. Alright, let's go more into this photon phenomenon, if you will. We've seen what happens when an electron absorbs a photon. However, what are the actual effects of this? One of two things happens to the energized electron, largely depending on the atom and its surroundings. It could return back to its ground state, or its normal condition, as its increased energy dissipates as heat and or as an emission of light of a longer wavelength than the absorbed light. This is known as fluorescence, and this is shown in this picture here. You see this emission, fluorescence emission. Now, what could also happen is that the energized electron actually leaves the atom and is accepted by an electron acceptor molecule, which becomes reduced. Now, remember, reduction describes when a molecule gains electrons. This really only happens if viable electron acceptors are available for the atom. So you can see this process in this picture here. It gets hit by the photon, gets excited, runs to the electron acceptor. This second possibility is critical in photosynthesis, as a sort of chain of electron acceptors passes energized electrons down in the reaction pathway. Now, in order to help differentiate between the two possibilities, we can go back to the slapping analogy. The first possibility sort of represents the electron as more of a mature adult. He gets slapped and gets angry, however, he lets off his steam or energy and goes back to his normal state. On the other hand, with the second possibility, the electron is more like an angry child that runs to his or her mom to get a hug, with the child not letting go of his angry energy. Now that we understand a bit more about light, we can start to look at the biological structures of photosynthesis that use such light. One of these structures is the leaf. Leaves are important to look at because they contain and support chloroplasts, a plant cell organelle. We will look at chloroplasts more in depth shortly, but first we need to look at leaves closely. Here we're going to look at a cross-section of a leaf. It has many things labeled, however, we're only going to talk about a few of these structures. Now, chloroplasts are mainly located in mesophyll cells. And the mesophyll, as you can see here in this diagram, is a layer of the leaf that has many air spaces and also has a high concentration of water vapor. The mesophyll is really like the cream of the donut, it's really the crucial component of the leaf, it being the photosynthetic tissue. Gases are exchanged between the interior of the leaf and the outside through microscopic pores called stomata. A singular stomata is called a stoma, by the way. The stomata are sort of like the gates of the leaf that open to allow carbon dioxide, CO2, in and oxygen, O2, out. H2O, water, is carried to the mesophyll of the leaf through veins, also pictured in the diagram here. In this way, veins act sort of like a highway for the water in the plant, transporting water where it needs to go, and it does need to reach the mesophyll in order to get to the chloroplasts where water is needed for photosynthesis. Speaking of chloroplasts, now that we've examined the leaf more closely, we can also do the same with chloroplasts to help us understand why they are so critical in photosynthesis. So we're going to use this diagram here to look at the different components of a chloroplast. Chloroplasts are enclosed by an outer and inner membranes, as you can see here in the diagram. In this way, they are similar to mitochondria, another organelle you might be familiar with. The inner membrane encloses a fluid-filled space called the stroma. The stroma contains most of the enzymes needed to make carbohydrates, the ultimate end product photosynthesis is trying to create. Remember that enzymes are proteins that help facilitate chemical reactions. Like the mesophyll, the stroma is really the cream of the donut, the part of the chloroplast that holds all of the richness. This includes membranous structures called thylakoids. They are these flat disc-like sacs shown here in the diagram. They sort of look like coins, and they really are the currency of the chloroplast. The chloroplast needs to have them to carry out photosynthesis, and they are what make the stroma, the cream, rich, quite literally if you think about coins, although I probably wouldn't like to have cream with metal coins in it. Anyways, the thylakoid membrane encloses a fluid-filled interior called the thylakoid lumen, which you can see here more clearly, the membrane and the lumen. And this can be analogized as the metal coat and interior of a coin. In some regions of the chloroplast, thylakoids are also stacked up in things called grana, singular granum, as you see here in the diagram. And they look just like a stack of coins, following our coin analogy. Also, you'll see that some thylakoid membranes actually extend from one granum to another, as you see here in the diagram. These extensions are called stroma lamellae. They look sort of like metal beams between the grana, right? Now, going back to the thylakoid membranes, it's key to point out that they really are the stars of the show in the chloroplast and of photosynthesis. They hold a great number of functions and purposes. One of those is to house the protein that makes ATP, the energy currency for the cell. So they're sort of like an energy factory in that sense. Thylakoid membranes also contain the pigments necessary for the light capturing reactions of photosynthesis. And this connects well because in the next section here we're going to talk about more about these pigments, what they are, and why they are critical in photosynthesis. So in this section we're going to talk about photosynthetic pigments. And what is a pigment? What is that? Pigments are simply substances that absorb visible light. Different pigments absorb different wavelengths. I think of pigments sort of like gears in that they are both activated by energy, and different gears may be activated by different types of energy or different circuits. Moreover, pigments drive processes like gears. Now, chlorophyll, which is this crazy molecule you'll see here, is the main pigment of photosynthesis. It absorbs primarily blue and red light, and if we go to the next slide, you can see that here. It does not absorb green light, so green light that hits plants often gets scattered or reflected. So this is why we see plants as green in color. Now, there are two kinds of chlorophyll. You have chlorophyll A and chlorophyll B. Chlorophyll A is the most important, as it initiates the light-dependent reactions of photosynthesis. Chlorophyll B is more of an accessory pigment that plays a more minor role in photosynthesis. The two pigments differ in minor changes in their chemical structure. Going back to our gear analogy, this can be manifested as two different types of gears. Chloroplasts also have other accessory photosynthetic pigments, carotenoids being one such group of them. You can see a whole bunch of them here on the right. Carotenoids are yellow and orange, showing how they absorb different wavelengths of light than chlorophyll. This expands the spectrum of light that can provide energy for photosynthesis. Why did I say provide energy? Well, chlorophyll can be excited by light either directly passing through it or from energy passed to it from accessory pigments that have been excited by light. Excited carotenoids can transfer their energy to chlorophyll A. You can think of carotenoids as little mini gears attached to big gears of chlorophyll A that help it move along slightly as an extra power source. Carotenoids also are antioxidants, which inhibit oxidation or the loss of electrons. So carotenoids inactivate highly reactive forms of oxygen generated in the chloroplasts from photosynthesis. Okay, so all that we've gone through so far is really the critical background knowledge that helps us understand the complex pathways of photosynthesis, which is what we're going to get into now. The process of photosynthesis can be separated into two separate parts, the light-dependent reactions and the carbon fixation reactions. Now we're going to go into the first, the light-dependent reactions, and see what they are all about and their role in photosynthesis. Things are going to get pretty complex here as I go into these pathways. However, hopefully I can express this material in a way you can easily understand it. So let's get started. So before getting into the nitty-gritty, it's often helpful to understand where we're going. What is the final product we are trying to achieve with these reactions? Well, the two key compounds that are produced in the light-dependent reactions are ATP and NADPH. Essentially, the light energy that chlorophyll captures is temporarily stored in these compounds for use in the carbon fixation reactions. ATP, or adenosine triphosphate, is produced through the phosphorylation, or the addition of a phosphate group, of ADP, adenosine diphosphate. And that makes sense, right? We're adding a phosphate group to ADP, which has two phosphate groups, hence di, making adenosine triphosphate. We're also producing NADPH from NADP+. NADP+ gets reduced, forming NADPH. Now you're probably like, where did this hydrogen come from? We just added a new element to this compound. Well, hydrogen essentially acts as an electron carrier for reduction. Remember, hydrogen is just a proton and an electron, and because it's much more difficult to transfer electrons solely because they are very unstable, hydrogen acts as an electron carrier that can be used to transfer these electrons. It's kind of like an electron taxi, right? So NADP+ is reduced as it interacts with two hydrogen atoms, and as the compound gains one proton from one hydrogen bonding to it and two electrons from two hydrogens that donated those electrons, we're left with neutral NADPH. So you can pretty much see these products being formed in this chemical equation here with a variety of reactants. And we're going to really see how water, light, and chlorophyll are really the key players here driving the process, while the other reactants just get transformed. Okay, so now you know that where we're going. Let's go into much greater detail behind this chemical equation and how it happens. So a critical component of the light-dependent reactions you need to understand are photosystems. Photosystems consist of a variety of components, which I'm going to lay out here. So light-dependent reactions initially begin when chlorophyll A and or accessory pigments absorb light. These pigments are organized into things called antenna complexes, which also contain pigment-binding proteins in the thylakoid membrane. The pigments and proteins are arranged in highly ordered groups that include about 250 chlorophyll molecules associated with specific enzymes and other proteins. Now, each antenna complex absorbs light energy and transfers it to a reaction center, which is a structure that consists of chlorophyll molecules and proteins that participate directly in photosynthesis. Now, what does that mean? Let's take a look at a picture to help us better understand all of these structures and photosystems in general. What we're looking at right here is a photosystem. Each of these circles are chlorophyll molecules that are organized into an antenna complex, which is essentially all these molecules all together. Now, when a chlorophyll molecule absorbs a photon from light, the energy is passed between the molecules in a process known as resonance in order to reach the reaction center, which, as we said, consists of chlorophyll molecules as well. Now, the energy that reaches the reaction center energizes or excites an electron that leaves the reaction center to be accepted by an electron acceptor, right? And we talked about this process way back in the beginning with the electromagnetic spectrum. If we want to analogize this process, we can go back to our slapping analogy from back then. Basically, the photon gives directions to these chlorophyll molecules in the antenna complex. They're like, okay, you got to slap someone. Someone's got to get slapped somewhere because I'm giving you these instructions. These instructions pass down the antenna complex to the reaction center, where this chlorophyll molecule is like, all right, got to slap someone, and bow, slaps the electron in the reaction center. This electron doesn't appreciate it one bit and leaves entirely to go get hugged by an electron acceptor. And this water down here is just being split and giving an electron to replace the one lost. Okay, so this process that occurs in photosystems is a critical step in the light-dependent reactions. Let's take a look at the specific photosystems involved in the light-dependent reactions. Two photosystems are involved in photosynthesis, photosystem 1 and photosystem 2. I know, not very creative. Now, what's different between them is their reaction centers that have chlorophyll A molecules with slightly different absorption spectra. Chlorophyll A molecules in photosystem 1 have an absorption peak of 700 nanometers, so they absorb best light with wavelengths of 700 nanometers. And because of that, the reaction center of photosystem 1 is commonly referred to as P700. Photosystem 2's chlorophyll A molecules have an absorption peak at 680 nanometers. They absorb wavelengths of 680 nanometers best, and therefore its reaction center is referred to as P680. Using our pigment gear analogy, P700 and P680 are just two reaction centers made of slightly different gears. So these two reaction centers play key roles in the light-dependent reactions. Okay, so what you're looking at here is the full light-dependent reactions pathway, assuming a non-cyclic electron transport, which we'll get into in a second, but first it's important to understand all of this craziness here. Okay, so we're going to go step by step through this, going into a bit more detail than this diagram shows. First we have photosystem 2, that gets activated as a pigment molecule in an antenna complex absorbs a photon. The photon, or the light energy, gets transferred to the reaction center, P680, which causes P680's electron to get excited. This energized electron is then accepted by a primary electron acceptor, and then it gets passed along an electron transport chain here and is donated to P700 in photosystem 1. Now, this passing along an electron transport chain causes some stuff. However, we're going to go back here for a second. Since P680 donated its electron, it becomes an ion, right? It becomes P680 plus because it lost negative charge. P680 plus is a very strong oxidizing agent, meaning that it causes other molecules to lose electrons very easily. This is what is happening with this water here. And we saw a bit of this earlier, right? This process is known as photolysis. P680 plus is pulling the electrons away from water. And this water gets actually split by an enzyme that breaks it down into 2 electrons, 2 protons, and oxygen. The 2 electrons go to P680 plus, making it lose its positive charge. The protons get released into the thylakoid lumen. And because oxygen does not exist in atomic form, the oxygen produced by splitting an H2O molecule is written as 1/2 O2. In order to actually get one oxygen molecule, you need 2 water molecules to be split. However, this isn't the case here, so that's why it's 1/2. So by this process, H2O is really the ultimate electron source, as it continually replaces the electrons here in P680. This is the source of almost all the oxygen in Earth's atmosphere as well. Now let's go back to the electron transport chain here. An electron donated by P680 is passing down this chain. As it gets passed down, it continually loses energy. You can kind of think of the energy level as a sort of staircase, it lowers every step of the way. Some of this energy that is released is used to pump protons, this H+, pump protons across the thylakoid membrane from the stroma to the thylakoid lumen. So this makes a proton gradient that powers a process to produce ATP from ADP by chemiosmosis. However, we're going to discuss this later. Just know that as this electron from P680 travels to P700, protons are continually being pumped by this structure here from the stroma to the lumen. Okay, so eventually, this electron being transported reaches photosystem 1. Now, photosystem 1 absorbs photons of its own. This causes an electron in P700 to get excited and be ejected, being accepted by an acceptor molecule, and then going into its own electron transport chain here. The electron that arrives from P680 just replenishes the electron P700 lost here when it absorbed a photon. Ferredoxin, which is a iron-containing protein, takes in the electron that's in this electron transport chain, transfers it to NADP+ in the presence of an enzyme called ferredoxin NADP+ reductase. Now, keep in mind that while single electrons pass through electron transport chains, 2 electrons are needed to reduce NADP+ to NADPH. So this reduction only happens when 2 electrons arrive. And so here, finally, NADPH, our desired molecule, is produced and released into the stroma. This cycle of electrons, starting from H2O and NADP+, continually occurs as long as light is present to kickstart the flow. Okay, so we have NADPH. However, we're missing something. Do you remember the other molecule the light-dependent reactions produce? ATP is what we're missing. How do we produce ATP here? Remember I said earlier how as the electrons get transported here, it loses energy. That becomes repurposed to pump protons across the membrane into the thylakoid lumen. This forms a proton gradient, and what that means is that there's an imbalance in the concentration of protons across the membrane. There's more protons here than over there. Moreover, even more protons get concentrated here as photolysis occurs, because this produces protons as a byproduct as well. Now, this proton gradient has a lot of free energy, because remember, molecules always want to go with their concentration gradient, meaning they want to establish an even concentration on both sides. So these protons really want to move over to the other side. However, the reason they don't just move across is because the membrane blocks them. However, a certain enzyme allows them to be able to cross this barrier. And you can see it here. It's called ATP synthase. As you can see, it's a transmembrane protein that goes into the stroma. Protons move through this channel, exerting energy that powers the phosphorylation of ADP to form ATP, which is then released into the stroma. This process is ultimately known as photophosphorylation, as the electron transport is what ultimately is the power source for this phosphorylation here. What's kind of interesting is that it's thought that as protons move down this channel, they actually cause conformational changes in this enzyme that causes this knob-looking thing to actually turn. So it's kind of cool. It's almost like a pepper grinder pumping out ATP. Okay, that in its entirety represents the light-dependent reactions assuming a non-cyclic electron flow. So I've mentioned this non-cyclic electron flow. However, there is such a thing called cyclic electron flow in light-dependent reactions, which I want to go through now. So this diagram shows the light-dependent reactions assuming cyclic electron flow. And it presents the material in a bit differently than the previous diagram. However, we can go through the same. Now, cyclic electron flow only includes photosystem 1. So this is why photosystem 2 is nowhere to be seen here. The reason the flow is called cyclic is because energized electrons originating from P700 eventually come back to P700. Imagine the electron as like a spoke on a bike. It goes through all the reactions necessary and then ends up right back where it started after a complete turn. What happens is that the photons still pass their energy right, and excite a p700 electron, which then goes on to an acceptor and enters a transport chain to stimulate photophosphorylation and chemiosmosis here, which used to be done in the electron transport chain before this with photosystem 2, but now it's done here. And also, the electron never goes to an NADP+. Instead, it just recycles back to photosystem 1 to enter the transport chain again. NADPH is not produced in light-dependent reactions utilizing cyclic electron flow, and water is never split, so oxygen isn't produced either. Therefore, the initial reactants would really only be ADP and phosphates, with the final products being only ATP. Now, it's important to note that photosynthesis could not be maintained if cyclic electron flow was only utilized. As we will see when we go over the carbon fixation reaction soon, NADPH is a necessary energy source. So you may ask, what is the very point of cyclic electron flow? And that would be reflecting the question on many scientists' minds right now. It's unclear exactly the significance of cyclic electron flow. However, there are a variety of theories, and there is a line of current research looking into it. A good reason a lot of scientists agree on is cyclic electron flow is adopted when there is a deficiency of NADP plus in the chloroplast. Though, as I said, more complex theories are being developed currently. So now we're going to move on to the second and last major step in photosynthesis, the carbon fixation reactions. The ATP and NADPH produced in the light-dependent reactions we just talked about are going to be used here. So let's dive in. So as we did with the light-dependent reactions, we're just going to take a quick look here at the carbon fixation reactions with its summarized chemical formula. The main end product we are making here, and the main end product of photosynthesis as a whole, is organic molecules, which most of the time takes its form as glucose, but can be another carbohydrate as well. CO2 is the main reactant we are interacting with, and we utilize the energy from ATP and NADPH produced in light-dependent reactions to power this process. So this chemical equation here has all the stuff I just talked about written in chemistry language, but the C6H12O6 is really that prize product. The prize reactant is CO2. The Calvin cycle is a major component of the carbon fixation reactions. It was discovered by Melvin Calvin, Andrew Benson, and others in the 1950s. And it consists of 13 reactions that occur in the stroma of the chloroplast. These reactions are divided into three main phases: CO2 uptake, carbon reduction, and RU-BP regeneration. So we're going to examine each of these three main phases and what occurs in them. So what you're looking at here is the complete Calvin cycle. We're going to go through each of these steps labeled here. They have slightly different names than what I use, but they're essentially the same. And we're going to go through all the processes that occur. So the first step is CO2 uptake. Here it's called carbon fixation. What happens here is that CO2 molecules, and remember these CO2 come from gases, enter through the stomata, right? This is atmospheric CO2. These CO2 molecules react with a molecule called ribulose biphosphate, or RUBP, as you can see here. This reaction is catalyzed by this thing, rubisco. It's an enzyme. So this forms the molecule you see here. Now notice how CO2 contains a single carbon per molecule, RUBP 5 carbons per molecule. So it makes sense that they culminate into a molecule with 6 carbons, right? Now this molecule is very unstable. It really can't be kept in the 6 carbon form. So what happens is that it immediately breaks down into 2 molecules of phosphoglycerate, or PGA. So essentially what has happened here in this phase is the carbon from CO2 has entered a carbon skeleton, or has been fixed into this new carbon structure. Now, the Calvin cycle as a whole is known as the C3 pathway because the initial production of the initial carbon fixation reaction is a 3-carbon compound, so C3. Plants that initially fix carbon this way are called C3 plants, and we're soon going to see different pathways. However, for now, we're just looking at the Calvin cycle, the C3 pathway. So that's the first phase, CO2 uptake. It just consists of that one reaction between CO2 and RUBP, forming these 3 carbon molecules. Next, we have the carbon reduction phase right here. 2 steps happen here. in which the energy and reducing power of ATP and NADPH are used to convert PGA molecules into glyceraldehyde 3-phosphate molecules, or G3P. Now, for every 3 CO2 molecules that enter the Calvin cycle, 1 molecule of G3P is made. This is eventually used in carbohydrate synthesis. In fact, 1 molecule of G3P is essentially half a sugar, and when a second molecule is produced, glucose or fructose is formed. And if you're counting carbons, that makes sense because the chemical formula for glucose/fructose includes 6 carbons. So if you have 2 3-carbon molecules, you end up with 6. And so that's our prize. That's it. We've produced the sugar the plant desperately wants. However, there's still one more phase to go through. There is the RUBP regeneration phase. Now, while some of the G3P goes on to make sugars, not all of it does. When the cycle goes through, you're actually left with these extra G3P seen here. So through a series of 10 reactions in this phase, these G3P molecules get rearranged into ribulose phosphate molecules, ribulose biphosphate molecules, using ATP and a bunch of other stuff. So this essentially regenerates the cycle. RUBP is made again, and so we can power the cycle again, react with CO2. So that's the full Calvin cycle. We started with CO2, ATP, and NADPH, and up with sugar, C6H12O6. You can sort of think of the carbon atoms here as beads. So it's kind of like a bead bracelet factory, if you will. You know, we're adding beads, we're breaking beads, we're modifying beads, ultimately to get a nice bead bracelet that is desirable. So now I'm going to talk about this phenomenon that happens with many CO2 plants called photorespiration. Now, when it gets very hot, plants often get faced with a difficult predicament. You see, a plant needs CO2, right, for the Calvin cycle, but it also needs to prevent water loss. Now, gas exchange happens in the plant through the stomata of its leaves. Remember, we talked about that way back in the beginning of the video. But on very hot, dry days, plants close their stomata because they leak water they desperately need for the light-dependent reactions through them. Once these stomata close, the CO2 in the leaf gets rapidly used up, with O2 getting accumulated in the leaf as they are a byproduct of the light-dependent reactions and now cannot get out of the leaf through the stomata. Now this causes some problems because O2, oxygen, actually competes with CO2 for rubisco. Remember the enzyme that is key in the Calvin cycle. And what I mean by competes is that if there's actually a larger concentration of oxygen in the chloroplast rather than CO2, oxygen actually interacts with rubisco itself and gets fixed with RUBP rather than CO2. This degrades RUBP and by association a lot of the other intermediate molecules in the Calvin cycle, with them often ended up as just CO2 or H2O. And so this whole process is called photorespiration. However, photorespiration does not produce ATP, as another process known as cellular respiration does, which you might know. And it also reduces photosynthetic efficiency greatly, as a lot of the intermediate molecules in the Calvin cycle are removed entirely. So the plant definitely wants to avoid this. It is not something the plant willingly wants to undergo. So scientists are not entirely sure why photorespiration is even possible, right? Like, why would rubisco interact with oxygen? And this is another developing area of research. This picture here helps illustrate what photorespiration actually, when photorespiration actually occurs as compared to the Calvin cycle. So as we said, when temperatures are high and there's much more oxygen than carbon dioxide, you get photorespiration. On the other hand, when temperatures are medium-low and there's much more carbon dioxide than oxygen, I don't know if I said more carbon dioxide here. More oxygen, less carbon dioxide is photorespiration. More carbon dioxide, less oxygen is Calvin cycle. So if we want to apply this to our bead bracelet factory analogy, essentially this would be like when there's a sudden single bead shortage, right? And the bead bracelet factory has to resort to complex, slower methods to make bracelets in order to get by. Now, you may be asking yourself, wait, there are plants that permanently live in hot, dry environments. You know, the first one that probably comes to your mind is cacti. How do they survive through constant photorespiration? Well, plants that live in these environments have special adaptations, introducing new chemical pathways in their mesophyll cells. There are two types of plants that have these adaptations, C4 and CAM plants. C4 plants first fix CO2, atmospheric CO2, to a special 4-carbon compound, hence C4, called oxaloacetate. CAM plants also do this, but only at night. So it's important to note these adaptations do not replace the Calvin cycle. They simply come before it. So in order to better understand what I'm talking about here, let's go more in depth with these C4 and CAM pathways. Let's first take a look at the C4 plants and the C4 pathway. Now, as we said earlier, the C4 pathway, in the C4 pathway, atmospheric CO2 is first fixed to a molecule called oxaloacetate. This occurs not only before the C3 pathway, but in different cells entirely. So let's look at this anatomy here of a C4 plant. Here it's showing corn, compared to a C3 plant. In a C4 plant, the Calvin cycle doesn't actually occur in a mesophyll cell like the C3 plant. Rather, it occurs in chloroplast-containing bundle sheath cells, seen here, that encircle the vein. The C4 pathway happens in the plant's mesophyll cells, so they act as a CO2 pump for the C3 pathway here in the bundle sheath cells. So I'm talking about the C4 pathway a lot. Let's look specifically into what that is and the reactions that take place here. What you're looking at now is a full diagram of the C4 pathway. So let's go through it. First, we have atmospheric CO2 that comes in from the stomata, right? This CO2 interacts with an enzyme that really is the star of the show of the C4 pathway called PEP carboxylase. This enzyme has an extreme affinity for CO2, even in low concentrations, and catalyzes the reaction between CO2 and a 3-carbon compound called phosphoenol pyruvate, or much more simply PEP. PEP carboxylase is like a super strong magnet, really, that attracts CO2 and ensures that this reaction will always take place even in environments with very low CO2. So this reaction forms oxaloacetate. After that, oxaloacetate is then converted to some other 4-carbon compound, usually malate, using energy from NADPH. This malate then leaves the mesophyll cell and then goes into the bundle sheath cell, where a different enzyme reacts with malate, yielding pyruvate, CO2, and NADPH, which isn't pictured here, but it's used to replace the one used earlier. The CO2 that is released then goes on to kickstart the Calvin cycle here, which ultimately produces sugars for the plant. The pyruvate formed goes back into the mesophyll cell, where it reacts with ATP to regenerate a PEP molecule. And so here you have this complete C4 pathway, this cycle. And this is really helpful for plants in arid conditions because the pathway captures CO2 very efficiently thanks to PEP carboxylase, which has a very strong affinity, as we said, for CO2. And so really, the C4 plants never experience photorespiration because the CO2 concentration in the bundle sheath cells, where rubisco is present, is always high. However, a downside of the C4 pathway is that overall it expends more ATP. This is worth it if you're a plant in arid conditions. However, C3 plants are favored at lower temperatures as they expend less ATP. Now, not all plants that live in dry conditions undergo the C4 pathway. Some plants undergo the CAM pathway instead, shown here, which is similar to the C4 pathway but with some significant changes. Firstly, CAM plants only open their stomata at night, which you would think is unusual, right? Because photosynthesis only occurs in the day when there's light. Why do the CAM plants do this? Why do they open their stomata now? Well, what CAM plants do is actually store CO2 throughout the night by first using the enzyme PEP carboxylase to fix CO2 to oxaloacetate, which is then converted to malate, and then stored in cell vacuoles or storage compartments. So they do that during the night, and then during the day, when the stomata are closed, the stored CO2 is removed from the malate through a chemical reaction, similar to the C4 pathway, and then the CO2 is readily available to be fixed into the C3 pathway. So we can kind of think of this pathway like when you plug your phone or your device in to charge at night. You know, throughout the night, the device gains all the necessary energy or required power to efficiently work in the day. And this is just like a CAM plant, right? It gains all its required CO2 at night so they can efficiently undergo the Calvin cycle, the C3 pathway, during the day. The benefit of a CAM pathway is that much less water is lost for the plant. Since they only open their stomata at night when temperatures are at their lowest, water loss is also at its lowest. This allows CAM plants to live in deserts where neither C3 nor C4 plants can live. So to finish off this section on the carbon fixation reactions, I've got this slide here just to compare the different pathways. So, with the C3 pathway, it's only the Calvin cycle, right? All these reactions occur in the mesophyll cells and expends the least energy, but suffers in hot climates, right? Because of a photorespiration, right? C4 pathway, CO2 is first fixed to PEP by this very powerful enzyme, PEP carboxylase, right? To eventually be stored to these 4-carbon mediums, right? The C4 pathway in mesophyll cells the C4 pathway occurs in mesophyll cells, right? Then the C3 pathway actually occurs in bundle sheath cells, right? So you have this pumping CO2 to the bundle sheath cells for the C3 pathway. The C4 pathway, C4 plants, are effective in hot climates, but expends more energy than C3 plants, so you don't really want to use it in moderate to low climates. And finally, the CAM pathway, the initial CO2 fixation is similar to the C4 pathway, right, with the oxaloacetate then converted to malate to be stored in still vacuoles. This only happens at night. Remember, stomata only open at night to let in CO2. And the stored CO2 can then be used, just like the C4 pathway, by the C3 pathway to produce sugars during the day. So to end off this video, we're just going to look at a quick summary of what we talked about, a summary of photosynthesis. So what's the gist of photosynthesis? We're using light energy captured by chlorophyll to synthesize carbohydrates. The very basic chemical equation, CO2, H2O, to form C6H12O6 and O2. Remember, the reactions are divided into two phases, light-dependent reactions in the thylakoid membrane and the carbon fixation reactions in the stroma. Light-dependent reactions, right? The light energy is converted to chemical energy. Chlorophyll captures this, causing these electrons to become excited. This is transferred to an acceptor molecule. It was replaced by an electron from H2O in the case of photosystem 2, right? This is photolysis, causing O2 to be a byproduct. Some energy of the energized electron is used to phosphorylate ADP to make ATP, and NADP plus becomes reduced as well from these electrons, forming NADPH. And these are these products of light-dependent reactions, ATP and NADPH, which are used to power the next phase, the carbon fixation reactions. So these molecules are not suited for long-term energy storage, so they need to be utilized to form carbohydrates, which is suited for that. So CO2 atoms, CO2 molecules, I should say, from the atmosphere are fixed to organic molecule skeletons in the Calvin cycle. Right, this is what directly produces sugars through the cycle. The C4 pathway is used for greater efficiency in collecting the CO2 for the C3 pathway through the PEP carboxylase enzyme. And the CAM pathway is similar to the C4 pathway, but is only activated at night. So that's pretty much it for this video. I have this works cited page here. A majority of the information for this video was taken from the AP Biology textbook, or this specific one here, Biology, with the main editor, Eldra Solomon. And that's it. Thank you very much for watching, and I hope you have a fantastic day.